Hello, everyone. Welcome to the Emerging Trends in Pharmaceutical and Biopharmaceutical Analysis. I'm Jerome Workman, the Executive Editor of Spectroscopy, and I'll be your moderator for this event. We are pleased to bring you this session, which is part of a two-day virtual symposium presented by Spectroscopy, the Society for Applied Spectroscopy, and the Koblenz Society. The Society for Applied Spectroscopy, or SAS, is a nonprofit organization dedicated to the dissemination of information related to spectroscopy. SAS is pleased to be the publisher of the internationally recognized peer-reviewed journal Applied Spectroscopy. The Koblenz Society is a nonprofit organization whose purpose is to foster the understanding and application of vibrational spectroscopy. Both societies have an educational mission, and this event is part of that mission. We invite you to visit both societies' websites to learn more. This virtual symposium is sponsored by CEM, PerkinElmer, Thermo Fisher, and Metrome, and Endress and Hauser. And now a statement from our sponsors. Today's event would not be possible without the support of our sponsors. CEM Corporation began manufacturing analytical instruments in 1978. In 1985, CEM developed the first microwave digestion system, revolutionizing sample preparation for trace metals analysis. Since then, continuous innovation has kept CEM at the forefront of sample preparation. With worldwide headquarters located just outside of Charlotte, North Carolina, CEM operates 7 subsidiaries as well as a large dealer network covering the globe. CEM truly is the global leader in microwave sample preparation. At Metrome USA, we're more than a leader in chemical analysis. We're a community driven by passion, integrity, and a commitment to quality and innovation. With expertise in both laboratory and process systems for titration, ion chromatography, electrochemistry, and spectroscopy, our approach goes far beyond just technology. Discover how our dedication to both science and humanity shapes everything we do at www.metrome.com. Customers worldwide trust Thermo Fisher tools and solutions to help them accelerate innovation and enhance productivity. Together, they are making advancements that truly matter. Thermo Fisher achieves this by providing an unmatched combination of innovative technologies, purchasing convenience, and comprehensive support. PerkinElmer is a global provider of scientific solutions with an 80-plus year history of bringing expertise, innovation, and technology to our customers. They manufacture the latest in analytical instrumentation and provide essential lab support services to streamline workflows. Partnering with scientists, clinicians, and lab technicians, they accelerate scientific outcomes and aid the development of groundbreaking products. PerkinElmer believes in the power of science towards a healthier, safer, and more sustainable world. Endress and Hauser provides advanced measurement and automation solutions for industrial processes across the globe. Their offerings include instruments for flow, level, pressure, temperature, and liquid and gas analysis. With a focus on innovation, reliability, and digital integration, they serve a plethora of industries. Their portfolio comprises of many products for any application, from basic to highly specialized, ensuring efficient and sustainable process control. We have a few housekeeping announcements before we begin. First, we encourage you to go to the resource section of our platform on the bottom of the media player. There you can find resources from our sponsors, as well as a link to view the other sessions of this event. We also encourage you to ask questions during the event today. You can submit questions by typing them in the Q&A box, which can be found underneath the media player. We will answer all questions at the end of today's event, after all talks have been presented. So please include the name of the speaker your question is for. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. We have a great lineup today, and I think you're all going to enjoy hearing from all of our speakers. Today's morning session will feature talks by Jürgen Popp, Patricia Alittner, Rohit Reddy, and Belinda Pastrana. I'm now pleased to introduce our first speaker, Jürgen Popp. Jürgen Popp studied chemistry at the universities of Erlangen and Würzburg, earning his PhD before completing postdoctoral work at Yale University. He returned to Würzburg for his habilitation in 2002 and has since held the chair of physical chemistry at Friedrich Schiller University in Jena. Since 2006, he has also served as scientific director of the Leibniz Institute of Photonic Technology. Popp is a global leader in biophotonics and optical health technologies, advancing research from basic photonics to clinical translation. He has authored over 1,100 peer-reviewed papers, over 43,400 citations with an H-index of 87, and holds 21 patents. Popp has delivered more than 200 invited lectures, including over 60 keynote or plenaries, and organized major scientific events such as the I-Course 2014. He is editor-in-chief of the Journal of Biophotonics and a key collaborator in numerous international academic, clinical, and industrial projects. Under his leadership, the Leibniz Center for Photonics in Infection Research, LPI, was selected for Germany's National Research Infrastructure Roadmap with substantial federal funding. A member of the Photonics 21 Executive Board, Popp has earned many honors, including the 2016 Pittsburgh Spectroscopy Award, the 2023 FACS Charles Mann Award, and an honorary doctorate from the University of Albany, SUNY. Jürgen, thank you for joining us today. Please get us started.
Yeah, ladies and gentlemen, it's a great pleasure to give today the presentation on Raman spectroscopy for on-site diagnostics and therapeutic drug monitoring in biopharmaceutical applications. My name is Jürgen Popp. I'm from the University of Jena, as well as from the Leibniz Institute of Photonic Technology in Jena. When we talk about drug monitoring, if we talk about on-site technology or point of care, we need to consider what are the guidelines in order to do point of care testing. What are the requirements for the point of care testing devices? Well, at the very end, the technique should be affordable. This means it should be cost-effective technology. It needs to be sensitive in terms of minimal false negative or being able for trace detection. It should be very specific. So this means minimal false positive or no crosstalks between different molecules. It should be user-friendly, and this means just minimal steps to carry out the tests should be required, and this means the entire analytical process needs to be somehow converted, and it needs to be rapid and robust. This means short turnaround time and no need for, let's say, any storage or long sample transport. It should be equipment free. This means no complex equipment should be needed, and it should deliver all this information directly to the user. Well, the technique we are using is Raman spectroscopy, and here you can also see one of our latest confocal Raman microscope, what we have developed. It's already miniaturized, just this size, and it's working the same way like the commercial ones. I will show you some Raman data later on, but let's come first to the working principle of Raman spectroscopy. So what you need is at the very end your sample. You need to shine your laser onto the sample, and what you will get is something like a molecular fingerprint information of the molecules within the laser focus. So we are looking at the inelastic light scattering. So we have to remove the elastic light scattering, the Rayleigh scattering, and then we can get really nice information on the molecules within the laser focus. But what we have to consider is when we are talking about biological samples, biomedical samples, then the constituents of those samples are mainly the same A biocell or a cell itself, it contains of water, proteins, small molecules, RNA, phospholipids, DNA, polysaccharides, and so on. And they don't differ too much. And therefore, at the very end, you need some tools like AI tools in order to do the analysis of those data. I'd like to give you some examples, and I'd like to start with the Raman-based in vitro diagnostics of infection. So when we talk about infection, then there are two ways we can look at the infections. One is, for example, to characterize the cellular host response, and this means we want to see what is the amount of the white blood cells, the different type of white blood cells, what is the ratio to each other, and are they activated or not activated. So this is what we call the host response. And the other way is to do this. You can also directly isolate the infection-causing pathogens, and when you have bacteria or something like this, then we get, for example, we can also look at the resistance profile. Due to the time constraints we have, I'd like just to talk about the host response using Raman spectroscopy. So just to clarify, what is the host response and what is our approach? So what we want to do is we want to do, first of all, a spectral phenotyping. Then we want to use this information to go for an in vitro diagnostics. And then certainly we have also to implement this into a clinical workflow. I will cover all these three topics, and let's start with the spectral phenotyping. So what does it mean? So when we look at a single cell or cells itself, so then we have the genes, we have the transcriptome, we have the proteome, in total the metabolome, and everything all together is somehow defining the phenotype. So when we do Raman spectroscopy of a single cell, for example, then we are looking at the phenotype. And this is what we are looking at the spectral phenotype. And what is it? It's nothing else than the entity of molecular interaction and structural features expressed within a cell or tissue at a certain time and under certain conditions. And in order to do the analysis, certainly we need some mathematics, some deep learning approaches. And then, for example, we can look at the white blood cells, at the spectral phenotype in a very sophisticated way, in a label-free way, and we can, so for example, easily separate that cells which has been, let's say, interacting with infections or with infection-causing pathogens, or with from the cells which has not been interacting with those. This we can easily separate. And to do so, as I said, we need to look at the data. We have to define the task. We have to do the experimental design. And if you are interested in, then you can look at this Nature protocol publication. And this is a comprehensive description of every step of the data analysis for biomedical Raman spectra. As I said, the experimental design, data preprocessing, machine learning, chemometrics, model evaluation, model transfer, external, internal validation. And if you are interested, you can also look at the software online. It's called the Raman Metrics software. Well, let's give you some examples. So when we do Raman spectroscopy on white blood cells, then, for example, the white blood cells, they consist out of neutrophils, monocytes, lymphocytes. In total, it's called the leukocytes. And we have always a certain percentage, and this is what we call, this is the diagnostic identification point of the immune phenotype. And if there is, for example, a shift in the population, this means a change in the amount of neutrophils versus lymphocytes and monocytes, then this is giving you some information about, like, the blood count, about your health status. And if there are some pathogens present, then we can also look by using Raman spectroscopy, for example, at the neutrophils to see is there any change due to this interaction. And we can even go for the systemic host response. I'd like to show you this later on a clinical study. So what we need is a small portion of blood, 500 microliter. We isolate the white blood cells. And then we have developed here a Raman device, a Raman high throughput device, which allows us to do the in vitro diagnostics in a very rapid way. So we can measure something like 90 cells per minute. And this allows us to measure in a short period of time something like 1500 cells, which is needed in order to do really the analysis of the blood count. And then we have, for example, published this device because it's somehow very special, because we don't take anymore these complete spectral resolution which you can achieve for Raman spectroscopy. Now we're going for 10 to 15 inverse centimeters. We have opened the slit. We have worked on the optimization. This was the way that we can really very fast. And what we do is we take an average spectrum of a single cell, so our laser focus is widened up to something like 8 microns in diameter. This allows us to be very fast. And this we tested, for example, on in different settings. For example, we looked at neutrophils and want to identify the host response when, for example, there are some bacteria present, gram-positive, gram-negative, or fungi. And we did this in a hierarchical way. First, we looked at the neutrophils infected versus non-infected, and there with a balanced accuracy around more than 92 or 93%, we can separate both. Then we can go for the infected. Then we can also, with a balanced accuracy of 92, separate between bacterial infection versus a fungal infection, just looking at the white blood cells, the neutrophils. And then even for the gram-positive, with a little bit less accuracy, we could separate gram-positive from gram-negative. Then in another study, for example, we were looking also at the virus particles, and we can learn something about, for example, the activation process of neutrophils. You need, for example, TNF alpha in order to really look at the host response when COVID or SARS-CoV-2 particles are present. And this we can also nicely separate, and we can also work, for example, from virus to virus. Also, again, on single cell level, we could easily also separate between different virus particles on the single cell level. So this is when we talk about host response, we can easily do this. So this is work which is going on for a quite long time, and just recently we were working also here in the emergency room, and here you can see we take this small amount of blood, then we do some pre-processing. Nowadays we can do this even faster. So this is then the Raman device, the high throughput Raman device, and then we do our measurement, and everything is now, when the samples is inside the Raman device, everything is done automatically. We measure up to 12 to 1500 cells, and then this allows us to do the identification. So these are now real samples coming from the emergency room, and each dot is a single person. We have measured something like 1500 cells. And the red ones were persons where we have a viral infection. The blue ones, people have a bacterial infection. And this has been then confirmed afterwards, because our process just needs 60 minutes. Just to summarize this, here you can see the entire process. So we have a patient which has been coming to the emergency room and has suspected an infection. We took this blood, 500 microliters, and then within 60 minutes we have this entire information. And what is quite important, this allows us, for example, now to identify these people have a viral infection, and then they need an antiviral treatment, or bacterial infection, they need an antibiotic treatment. And when there would be people with a fungal infection, then they need an antimycotic treatment. What is the underlying hypothesis, which has been proven with the spectroscopic information? We can lead to a diagnostic-driven therapy, and this much faster than classical microbiology or the clinical chemistry, because we can do this within 60 minutes or even less. Now I'd like to come to a study, a longitudinal study, where we looked at people having a bacterial sepsis versus healthy or people having a COVID-19 sepsis. Well, and here you can see the setting of the study. We have 75 people with a bacterial sepsis according to the definition 3, then COVID-19 associated sepsis and a healthy donor group. And it was a longitudinal study. This means we have T1 is in the acute phase. So this means 3 days after they have been, after the onset on a sepsis, when they are in the ICU, in the intensive care unit, or then this T2 was the subacute phase, and this was something 7 ± 1 days after the onset of sepsis. Time 3 was 6 months later, and time point 4 was 12 months later. And the first question was, can we separate the diseased people from the healthy one? And here you can see that with a very nice accuracy, balanced accuracy, we can do so. And this even after 12 months, we could say these people were healthy and these people had a sepsis. Then we asked our question, does it also work between COVID-19 and the healthy group? And also the sepsis group, you can see the numbers are almost the same. And then we also ask ourselves, can we separate also these two diseases from each other? And the numbers dropped a little bit, but still we were able to do so. And this already shows that Raman spectroscopy on a single cell level is a very powerful tool in order to do the analysis on the host response, and this in a very short period of time. We implemented also a clinical workflow from the Raman measurement. So, for example, the sample in the blood withdrawn was on the ward on the ICU. Then there was a short blood transport via this pneumatic tube, or in German Rohrpost, to the clinical chemistry. And then in the clinical chemistry, we had a complete workflow with this 500 microliter of blood. Then we have our Raman team doing the measurements, and we measured at least something like 15 to 2,000 cells per time point. And then the data analysis is being taken. The entire process is because this sample has to be transported to the clinical chemistry, is always less than 2 hours until we get the result. This is the Raman device, our miniaturized Raman device, and here you can see this with a comparison with a commercial one. This is human blood plasma. You can see we have a really nice spectral response. The red one is our point of care device, and now we are going for a series production of our new compact high throughput Raman device to do some multi-central studies. We will also bring one to Albany and one to Innsbruck, and then we are also working on the further development of the AI-assisted Raman diagnostics. Well, the second topic I'd like to share with you is how we can advance therapeutic drug monitoring by using Raman spectroscopy. And when we talk about therapeutic drug monitoring, why is this necessary? Because there's very often just a narrow therapeutic window where we have small variations, where we have the risk of underdosing or overdosing. So this is, for example, if we have this therapeutic range and we have then we are underdosing for a certain time, then we will have a low efficiency or efficacy. And if we are going above, then we might have a high toxicity. So this means we have to do a therapeutic drug monitoring, which allows us really almost a continuous monitoring. So this means we will really try to miniaturize side effects and toxicity by using those approaches. So Raman is not very strong. So this is the Raman intensity. And how can we increase the Raman response? Certainly we can go for high intensity, we can go for SIRS. This is something we are working on. We can also go more to the blue or to the UV. This is also something we are using, or we can go into electronic resonance, then doing resonance Raman spectroscopy. What I'd like to share with you is something which most people are not really taking care of. The efficient use of the presented molecules within your sample. Why? Because when we are doing Raman spectroscopy, we can also go for, so to say, for a fiber-enhanced Raman spectroscopy. What does it mean? And this we have been working for a quite long time. We have published many of those papers where we use, for example, something like a hollow core fiber in order to do Raman spectroscopy. And why is this of importance? I'd like to show this here once again, because when you are just using a microscope and you focus your laser into your sample, then you will just get the molecules which are in your sample. Yeah? And there's no way that you really can widen up the focus too much. But what you can do is you can use an optical fiber, as I said, a hollow core fiber. You can fill the hollow core fiber with this little droplet there, and then you can make efficient use out of your sample. And here you can see the approach. We have a laser, a spectrometer. We couple via our objective this laser light into the sample, into this optical fiber, and then via syringe pump, the sample is put into this hollow core fiber, and this allows us to do then nice measurement at very low concentrations. This is what, for example, those hollow core fibers looks like. If you are interested, you can also look at our homepage at the Leibniz Institute of Photonic Technology. We have an entire fiber drawing facility where we can produce those fibers, and if you are interested in those fibers, just get in contact with us. So this is here, for example, an air-clad fiber with bigger holes, which can be used easier compared to PCF fibers. And here you can see the advantage. If we look, for example, here at this antibiotic cefoxiam in a cuvette or in this fiber-enhanced Raman spectroscopic approach, you can see we can have a nice increase in the signal-to-noise ratio. And what is quite important, those fibers, these hollow core fibers, have also a nice transmittance from the UV to the NIR, and this means we can also use different wavelengths. We can even combine resonance Raman with, for example, this fiber-enhanced approach, because we have this nice light guidance. Just to show you, here some results of cefoxiam, first with synthetic data. You can see we have a nice linear dependency, and if we go, for example, now to human urine, and this was then a study by a PhD student, and this is the calibration curve in red with the black points, and the blue ones are coming 7 hours or 11 hours after a single dose of cefoxiam was given to the student. Then you can see this nice agreement between what we have found via our calibration curve and what has found by the Raman spectroscopy. And what is nice, we have a nice limit of detection compared to conventional Raman spectroscopy, and we are in the complete diagnostic range what is needed in order to do those analyses. And this we can more or less do continuously. Well, now I'd like to shift the gears a little bit and go into on-site intraoperative cancer diagnostic and therapy by in vivo in vivo multimodal imaging. So what is the idea behind this? So if you have, for example, a tumor, this is here you see an image, an endoscopic image, and in the circle you see something, and this is the tumor, for example, then you have just white light information to see where's the tumor border and so on and so forth. What we want to use is now optical molecular imaging, for example, coherent anti-Stokes Raman scattering, which is essentially like Raman, but now this is a nonlinear process. It's a Chi3 process, and we can now coherently excite these molecules. This allows us to be much more intense and therefore more faster in terms of getting the data by using this CAS approach. And this we can go for chemicals like lipids, proteins, RNA, DNA. And since we are using pulsed lasers, we get additional information. For example, we can get information from 2-photon fluorescence. We can look at elastine and NADPH, for example, but we can also do fluorescence lifetime imaging, 2-photon fluorescence lifetime imaging. Then we can also go to the cellular metabolic states, and we can have really nice, we can go for metabolic imaging. And In addition, we get also, for example, information from tubulin or collagen by SHG. Whenever there are molecules having no central symmetry, they will give us a SHG signal. So all this information we can collect easily together. We did a case study, for example, on a human lymph node, and you can see on the right side the image is showing the lymph node. It's something like 1.5 to 1.8 centimeters. On the left side, you can see the SFG, the sum frequency generations. You can see SHG, you can see the fluorescence, but you can also see the cars from the adipocytes. And this is the overview, but we can nicely zoom in as well. And this means we can overlay and zoom in and get here subcellular information. And what is also quite important, we want to translate this knowledge, this multimodal image knowledge, into something the physician can easily understand. And therefore, we are using, for example, deep learning approaches without going into the details. For example, here, this generative adversarial networks we can use in order to translate our multimodal image into an HE stain. And this allows us then, for example, to do the calculation of, for example, here we have this multimodal image of the lymph node. We can zoom in, we can do the calculation, we can produce an HE stain, we can zoom in, and this information on the right side can easily be understood by the physician, whereas the multimodal image is not. And this is the way we want to go, because we want to go for in vivo diagnostics and therapy. And this means we want to look at the tumor. And you can now see, for example, this multimodal image. We translate this into an HE stain, and this can be easily now understood. A picture is worse than 1,000 words. And here, this is the idea behind what we want to achieve in the future. In order to do this and to achieve this, we have developed here a nonlinear endo microscope with the company Grintech. We have a couple of one fiber for laser delivery. We have a scan head with galvo scanning systems, and we have then two fibers in order to get the signal out, one for the nonlinear imaging and one, for example, for ICG fluorescence. And then we have just PMTs, what we use in order to detect these couple of signals. Just to show you what it looks like. So this is within a cartridge. It's not really large, a lot of air around. Down here we have our endomicroscope, we have our scanning imaging laser, and this is the electronics and the detection unit. And with this, we'd like to move quite soon in the operation room with our rigid approach and going for head and neck cancer and for brain cancer diagnostics. We did already some smaller studies in order to learn what we can achieve. And this was here, for example, on head and neck cancer. And this is a specimen, again, something like 1 centimeter. We have first to correct for uneven illumination. And then we have our corrected image. Then when we have our corrected image, then the slide is being stained, for example. Then we have our HE stain. The physician, the pathologist is doing the annotation. And then we have our annotation mask put onto this image. And then we have our resisted image. And then we use, for example, here some deep learning approaches here, this semantic segmentation, where we use, for example, for training the white cells of a chessboard and the black one for testing. And if we go, for example, for the ground truth and to the prediction, and we go for 6 classes, we have an average specificity around 98%. The sensitivity is quite low. But if you just go for 2 classes, healthy versus diseased, then you can see we have already an increase in sensitivity. And there we believe that this might be a future technique. Well, what is nice, if you can go for diagnostics, why not also using light for the treatment? And this is what I'd like to show you last. And here you can see that we can easily couple in also a second laser, a CPA laser, a femtosecond laser working at 1032 nanometers, and this we use for ablation. Ablation is really not something new, because it has been used, for example, in LASIK, in dentistry, in vocal folds treatment, or neurosurgery. What is quite important, we have subcellular precision, we have low collateral damage, and we have a high speed scanning, and this allows us to go for diagnostic and selective tissue removal. And this I'd like to show you on one example here, see and treat image processing driven ablation. This is a brain sample from a human person, and here you can see there are some cholesterol, and this was before ablation. So what we can do here is, first I'd like to explain you the procedure. First we take a multimodal image. When we have done this multimodal image, then we go for a k-mean clustering. Then we can say, oh, we have here these clusters, this cholesterol inclusions, and this cluster we want to remove. Then we have our raw ablation mask, then our filtered ablation mask, and then we do the ablation. And you can see that we can easily remove this. But what is fantastic, we can do this in real time. And this I'd like to show you here with this little movie. And when we start, then we take the first image, then the processing starts, and then when we have our annotation mask, then we can go for diagnostic and therapy at the same time. Yeah, so we scan and we remove at the same time, and you can see this here in real time. And this makes it quite unique, and this we would like to push further towards then clinical applications on, for example, head and neck cancer, but also on neurosurgery. Yeah, with this, I'd like to thank my colleagues, my co-workers, for this very enthusiastic team and very much engaged, pushing always the limits towards new boundaries. I'm very much grateful for funding coming from various organizations. Many thanks also to our collaboration partners. And if you want to learn more on those topics, there will be the ICOP, the International Conference on Biophotonics in Albany next March. If you are interested, then this is not a purely scientific conference. It's also about how can we implement those new findings into the clinic, into the real world applications. With this and an image of Jena, I'd like to close, and I'd like to thank you very much for your attention.
Thank you, Jürgen, for that excellent talk. I would now like to introduce our next speaker, Patrice Leitner. She's an analytical chemist specializing in microscopy and spectroscopy with an emphasis on particle characterization. Her career spans multiple industries, beginning with analytical support for coal and nuclear power plants. She later advanced to senior research scientist at a leading microscopy consulting firm, concentrating on electron microscopy. Patrice then joined a major pharmaceutical company where she focused on forensic particle characterization and failure analysis across manufacturing and engineering teams. Currently at Pfizer, she assesses particulate matter in biological product development using advanced microscopy and spectroscopy. She also leads the Pfizer Particulate Characterization Network and the Biotherapeutic Particle Common Interest Group, promoting collaboration and innovation in the field. Patrice, thank you for joining us today. You have the floor.
Hello, my name is Patrice Leitner, Senior Scientist in Particle Characterization at Pfizer. I work in Biotherapeutics Research and Development Group. Today I'll discuss how Raman spectroscopy helps us understand silicon and protein interactions in biotherapeutics. My expertise spans particle isolation, microscopy, scanning electron microscopy, FTIR, and Raman. I am fortunate to have a career that involves combining the results from all of these techniques to solve problems. Today, I'm excited to share insights from our recent work using Raman spectroscopy as it relates to biotherapeutic R&D. Here's our roadmap for today's talk. We'll start with an introduction to protein pharmaceuticals and syringes, and then discuss why studying silicon-protein interactions is critical. Next, I'll introduce orthogonal techniques that have previously been used in our department to study silicon-protein interactions and their limitations. We'll then explore how Raman is used to study silicon and protein, followed by a look at silicon in syringes and experimental data. Finally, I'll wrap up with conclusions and future directions. Let's begin. Biologics have evolved significantly to meet patient needs, and with this evolution, new therapeutic modalities have emerged, such as antibody-drug conjugates, bispecific antibodies, and fusion proteins. These innovations have increased the complexity of formulation development, often requiring the creation of high concentration or complex formulations to achieve the necessary drug efficacy. As a result, it's essential to identify critical physiochemical liabilities early in the development process. These include challenges like protein aggregation, precipitation, opalescence, particle formation, and high viscosity. Addressing these factors early help ensure the safety, stability, and effectiveness of biological drugs as they move from development to patient care. Prefilled syringes offer several important advantages in the delivery of biotherapeutic drugs. First, they enable precise dosing, which is critical for patient safety and treatment efficacy. Their design also makes administration easier for both healthcare professionals and patients, supporting fast delivery times compared with an IV infusion and improving overall convenience. Another key benefit is the reduced chance of microbial contamination, since the drug is sealed in a sterile environment until use. Let's now look at the different syringe types available for the pharmaceutical industry. This slide compares syringe types and how the silicone oil is applied. There's glass syringes with a sprayed-on silicone, and they are widely available, but may lead to more interactions between silicone oil and the drug. There's also glass syringes with a baked-on silicone, so they have less free oil and improve stability, but availability can be limited. There's also COC syringes, which use a cross-link silicone coating, further reducing the free oil and improving stability, though they are a bit more expensive and less common. Choosing the right syringe type is important for minimizing unwanted interactions and ensuring drug stability. So why study silicone oil protein interactions? Protein aggregates and protein silicone aggregates may cause an increase in immunogenicity. Silicone oil droplets may act as adjuvants, potentially triggering unwanted immune responses. However, it is important to note that no increase in immunogenicity or safety issues have been observed from prefilled syringes currently on the market. So what do we know? We know that hydrophobic proteins tend to interact with silicone oil, particularly at the silicone-water interface. Agitation increases this aggregation, but it can be mitigated by surfactants like PS80 and PS20. What don't we know? There's still open questions about the influence of excipients on silicone protein interactions and how these interactions might change the secondary structure of proteins. These unknowns highlight the need for further research. And understanding these interactions helps us design safer, more stable formulations and anticipate potential risks in biotherapeutic development. Next, I'll introduce the orthogonal techniques we have used to study these interactions. Now that we've discussed why silicone-protein interactions matter, let's look at the analytical toolbox we use to study these interactions in detail. In our laboratory, we rely on several orthogonal techniques to get a comprehensive view of how proteins and silicone oils interact in biotherapeutic formulations. The three main methods are fluorescence microscopy, flow imaging, and flow cytometry. I'll walk through each technique in detail on the next slides, showing how they complement Raman spectroscopy and help us build a complete picture of silicone protein interactions in biotherapeutics. Let's start with fluorescence microscopy. In these experiments, we've used Bo-Dippy dye, which binds non-specifically to hydrophobic regions of both silicone oil and protein, and proteostat, which binds specifically to protein aggregates. The images on the slide compare samples with and without PS80, showing differences in protein absorption and aggregation. At the initial condition, protein absorption to the silicone oil can be observed in the absence of the surfactant PS80, but not over here with the PS80. So this highlights how surfactants can influence the interactions between proteins and silicone oil droplets. And while fluorescence microscopy is useful for visualizing these phenomena, it has some limitations. It examines only a small population of particles. It requires the addition of a chemical dye, and it can sometimes be a weak phenomenon and difficult to see even in a dark room with no ambient light. And remember, BODIPI binds non-specifically to hydrophobic regions in the protein as well as the oil. And this method provides no molecular information about the particles. Next, I'll discuss flow imaging, which provides a method of looking at larger particle populations. As you can gather from its name, flow imaging captures images of particles in a liquid as they flow past a camera. Silicone oil droplets typically appear as spherical particles with high-intensity centers, while the protein aggregates or non-silicone oil droplets often have more elongated amorphous shapes, as you can see attached to these silicone oil droplets in these images. Now, these images can be described in numeric terms of circularity, aspect ratio, object intensity standard deviation, and object intensity maximum, collectively known as S factors. So prior to running your study samples, you run reference samples of silicone oil and protein aggregates separately within the formulation. And these are graphed in terms of these S factors to create a mathematical equation that sorts these two populations into silicone oil and non-silicone oil. And as we can see with the graph on the right, the presence or absence of surfactants, such as PS80, can significantly influence the distribution of silicone-like and non-silicone-like particles. With PS80, we observe a greater count of the silicone oil droplets, while without it, there's an increase in the non-silicone-like particles. We also observe an increase in both types of particles in the aged samples. So while flow imaging is powerful for morphological analysis, it does have its limitation. One is that it only provides that morphological information, not molecular composition, and particles smaller than 5 microns are not imaged clearly. And also, this method does not provide information on particles that have both the silicone and protein components. So flow imaging is used to distinguish between silicone oil droplets and protein aggregates or non-silicone oil droplets based on the morphology, helping us to better understand their behavior and formulations. Next, I'll discuss flow cytometry, which combines fluorescence and flow imaging. As I just said, we're going to now look at flow cytometry. It's a powerful tool that lets us rapidly analyze large populations of particles in solution. And this combines the principles of fluorescence microscopy and flow imaging, so we can distinguish between silicone oil droplets and protein aggregates. In our experiments, we used the two dyes as we did in fluorescence microscopy, both the BODIPI, which remember binds nonspecifically to hydrophobic regions found in silicone oil and protein, and proteostat, which specifically labels protein aggregates. As particles pass through a laser beam, a bright field image and the fluorescence signals are measured, allowing us to quickly sort and quantify different particle types. One challenge is that the interaction between silicone and protein often lies close to this dividing line between the populations, making it difficult to distinguish them with absolute certainty. However, flow cytometry serves as an excellent screening method to identify samples with many particles near the interface, which can then be further studied using fluorescence or Raman spectroscopy for more detailed analysis. While flow cytometry is powerful for rapid screening, it does have limitations. It requires the addition of the dye, and one of those dyes being BODIPI, which binds non-specifically to hydrophobic regions even on the protein. It also provides no molecular information about the particles. And the information on particles that have both protein and silicone components is limited. Flow cytometry is best used as a high-throughput screening tool to identify samples with interesting populations, which can then be examined in more detail using other techniques. Next, I'm going to shift to how we use Raman spectroscopy to study silicone-protein interactions in solutions, building on the insights that we have gained from these orthogonal techniques. Okay. Let's jump into studying silicone-protein interactions in solution by Raman spectroscopy. I'd like to give a brief introduction to Raman spectroscopy for those of you who may not be familiar with the technique. It's a powerful analytical technique that allows us to study the molecular composition of samples by measuring how the laser light interacts with those molecules. So when a laser strikes a molecule, most of the photons are scattered elastically, but a small fraction, around 1 in 10 million, can undergo inelastic scattering, either gaining or losing energy from those molecular bonds. And this is going to produce a Raman spectrum, where the x-axis is the Raman shift in inverse centimeters, and the y-axis is the counts. One of the major advantages of Raman spectroscopy with a microscope module is its ability to analyze particles as small as a micron. It can analyze particles in liquid samples as well. The confocal nature of the technique also enables analysis through glass, making it ideal for studying particles in syringes or other containers. However, there are some limitations to keep in mind. The laser can potentially burn a hole through sensitive samples. Yes, I've done that. And fluorescence from the sample can sometimes inhibit the Raman signal. Overall, Raman spectroscopy provides unique insights into the composition and interaction of particles, making it an essential tool in biotherapeutic research and development. Before we dive into the details of the silicon-protein interactions and start looking at some data, it's essential to discuss how we've optimized our experimental conditions for the Raman spectroscopy. And this process starts with an understanding of the components involved, both in our method and what our samples look like. So for the method, we're going to consider factors like water and quartz, which can introduce background signals. And for the samples, we're going to focus on silicone and protein, the key players in our study. Mapping optimization is also critical. We're going to carefully adjust laser power and collection time to ensure we get reliable, interpretable spectra without damaging sensitive samples. By systematically optimizing these parameters, we can confidently distinguish between signals from silicone, protein, and any background interference. This foundational step sets us the stage for accurate mapping and the analysis of interactions in biotherapeutic formulations, helping us draw meaningful conclusions from our Raman data. Let's look at the spectrum for water. Notably, we see this rapid increase in counts after around 3,000 inverse centimeters, and this small broad peak at 1643. Now let's look at the Raman spectrum of a quartz cover slip placed on a mirror-finished stainless steel slide with water in between the slide and the cover slip. The goal here is to identify any interferences that might arise from adding that quartz cover slip that we're going to use to flatten our samples. The cover slip used here is approximately 0.15 to 0.18 millimeters thick. And we can see this large broad peak at 450 and some smaller peaks at around 600 and 800 inverse centimeters. Now let's look at one of our sample components, silicon oil, also known as polydimethylsiloxane, or PDMS for short. We can see this silicone methyl peak at 709, and this is the peak we're going to use to map the distribution and interaction of silicone within our samples. Our other sample component is going to be a protein solution. So the key peaks in this spectra include the amide 1 band, which is prominent around 1665, and is commonly used to assess protein secondary structure. But don't forget we have a small amount of interference with our peak that we saw in our water sample at 1645. The other notable features are the CH and CN stretching regions, as well as the spring mode at 1003. So as we go into our next couple slides, we're going to be looking at the spring mode, which is smaller, but a very sharp peak, and this amide 1 band, which is a larger peak, but is also a little bit broader. Let's get into the fun mapping data. This is an H protein drug, and the image at the top is from a sample with PS80, and the particle on the bottom is from a sample without PS80. And if we look at this episcopic illumination, we can see that elongated amorphous shapes that we typically associate with a protein aggregate. If we look at the map of the 709 peak, the silicone methyl bond, we see we can detect the presence of silicone in this particle. And remember, for the protein, we're going to look at the 1004 peak, which is that ring mode, as well as the amide 1 band at 1665. In this case, I'm seeing a better definition of the particle on the 1004 peak, and so that's the one I'm going to use going forward with mapping. Another thing to look at on both of these samples is that the top one was collected at a 50% laser power with 5 collections on each spot, And then on the bottom one was 100% laser power with only 2 collections. You can see we're getting pretty comparable maps for both of those. But the one thing to think about is with the 100% power, you may start burning holes through your samples, which is why you may want to go with the 50% power. But remember, these samples are in liquid, so sometimes they're moving, and we might only be able to capture it as it's going through the laser, so you might want to go with 100% power with the 2 collections. On this slide, we're looking at a circular particle imaged under the episcopic illumination, which we normally associate with silicone oil, alongside the Raman maps of the 709 peak, that silicone methyl peak, as well as the 1004 ring mode peak for protein. Now notice that clear separation in the map of the 709 peak, which indicates the presence and distribution of silicon within the particle. Now, in contrast, the map of that 1004 peak for protein does not show very strong separation. And actually, some areas in the protein map have background levels as high as the sample is itself. So based on these results, we would not conclude that any appreciable amount of protein is associated with this particular droplet. This highlights the value of Raman mapping for visualizing the spatial distribution of silicon and protein, and helping us assess their interactions and optimize analytical conditions for reliable analyses. In the future, we're going to start using flow imaging, including flow cytometry, to look at large changes in the population, and then start using Raman microscopy to start analyzing changes in the secondary structure. In the next slides, we're going to continue to explore how we've used Raman to monitor silicone coatings in syringes and track changes over time and location. So let's look at the Raman spectrum of the baked-on silicone coating that was removed from a syringe and analyzed in air. We have several peaks identified, including those for the methyl and methylene groups, some nitrogen, some oxygen, and then the silicone methyl groups and the silicone oxygen silicone groups. So while these are specific to silicone coating, what I want to concentrate on now is this peak at 2908, and that is because that is much larger than these other peaks down here in a much cleaner area, and we can use this to track changes over time and location. And this approach is especially useful when working through thicker glass walls or under time constraints, as the largest peak provides a reliable signal for monitoring. And by focusing on this key spectral feature, we can assess the uniformity and stability of silicone coatings in syringes, which is critical for ensuring the safety and performance of biotherapeutic products. On this slide, we're showing that we've mapped a short area of silicone coating inside of a syringe using Raman spectroscopy. The image over here on the left shows the coating under episcopic illumination, and the accompanying graph on the right signals the changes along those mapped regions. The spectra were collected at 10 micron steps. So this approach helps us to assess the uniformity of the silicone coating and actually can provide a fiducial mark to return to in future analyses of the syringe. So this slide shows how we monitor the stability of baked-on silicone coatings inside a syringe over time. This one is specifically held at 40 degrees Celsius. So using Raman spectroscopy, we track the signal intensity between 2880 and 2930 along the length of the syringe. We took measurements at different time points with the initial time at T0, T2 weeks, and T1 month. And each scan was taken at 100 microns. So these results indicate that there were no major changes in the thickness of the silicone coating over time, suggesting good stability under these conditions. Interestingly, one side of the syringe consistently showed a stronger signal, which corresponds to a thicker silicone layer. Let's wrap up the presentation with a few final thoughts. It's important to recognize the value of integrating Raman spectroscopy into our broader analytical toolbox. Raman allows us to analyze aqueous samples with minimal mechanical or chemical manipulation, making it possible to study particles, including droplets, directly in liquid environments. This technique is also well suited for non-destructive analysis of materials in syringes, enabling us to monitor coatings and interactions over extended studies, even within the same syringe. Raman spectroscopy can be combined with orthogonal techniques such as fluorescence microscopy, flow imaging, and flow cytometry. Together, these methods provide a comprehensive evaluation of biotherapeutic formulations, supporting the development of new modalities and container closures. By leveraging the strengths of each technique, we can more effectively address the challenges of formulation development and ensure the safety and efficacy of biotherapeutic products. So what's next? Our next steps are going to involve looking at changes in the secondary structure of proteins when they interact with silicone, and how these changes are influenced by different excipients, pH levels, and surfactants. So by studying these factors earlier in the development cycle, we can better inform formulation decisions and improve product stability and safety. We're also planning to go beyond silicone and explore protein interactions with a variety of excipients, including those currently in use and those that are still exploratory. This broader perspective will help us understand the full landscape of formulation challenges and opportunities. Ultimately, these studies will provide deeper insights that can guide formulation strategies, support regulatory submissions, and drive innovation in biotherapeutic development. Thank you for taking time to listen to my presentation today, and also from the support from Lindsay Crawford, Alex Langford, Michael Moore, Brad Colby, Michael Colby, Spectroscopy Online, Koblenz Society, and Society of Applied Spectroscopy. Thank you.
Thank you, Patrice, for that fascinating talk. As a reminder to everyone in the audience, you can submit a question by typing it in the Q&A box, which you can find underneath the media player. Also, please type in the name of the speaker your question is for. Questions will be answered at the end of today's session after all talks are complete. I would now like to introduce our next speaker, Rohith Reddy. Rohith Reddy is an associate professor at the University of Houston. He received his PhD from UIUC, BTech from IIT Madrid, and completed a postdoctoral fellowship at Harvard Medical School. He leads the medical imaging with the Lasers Lab, which develops advanced instrumentation and machine learning methods to tackle key biomedical challenges. His work has propelled spatiotemporal analysis of bone marrow disorders, Alzheimer's disease, kidney conditions, and gynecological cancers, and has been instrumental in establishing mid-infrared spectroscopic imaging for prostate and breast cancer diagnostics. His research centers on MIRSI and optical coherence tomography for biomedical applications. He holds 7 patents and has received numerous honors, including the FACS Innovation Award, the Thomas Hirschfeld Award, and the William F. Meggers Award from Applied Spectroscopy. He serves on the advisory board of Applied Spectroscopy and is president-elect of the Koblenz Society. Rohith, thank you for joining us today. You have the floor.
Hello everyone, thank you to Spectroscopy for inviting me to this Biopharma Symposium. Today I'll be talking about mid-infrared spectroscopic imaging for biomedical diagnostics. I'll start with a brief introduction to mid-infrared spectroscopic imaging and talk about its biomedical applications, and specifically I'll talk about how we can use machine learning for tissue classification and tissue subtype analysis, and how we can do cancer detection with this framework. And then I'll talk about some of the interesting and exciting new techniques that have been developed in the past few years. Specifically, I'll talk about photothermal infrared imaging and dual scope imaging, which address several different challenges with older methods and allow us to do much better in terms of biomedical diagnostics. And I'll give several examples of clinical and biomedical applications in ovarian cancer, bone fibrosis, and if we have time, lupus nephritis as well. In the end, I want to convince you that this technology is a very exciting field, and it has tremendous biomedical and clinical applications. The fundamental basis for infrared spectroscopy is the mid-infrared spectrum, absorption spectrum, and here is a typical tissue spectrum, mid-infrared spectrum of a tissue sample, and we can identify biomolecular constituents such as lipids, proteins, nucleic acids, and carbohydrates, which correspond to different wave numbers in the mid-infrared spectrum. Most of the information is actually here in what is called the fingerprint region of the mid-infrared spectrum, which is what we can use primarily for our analysis. And mid-infrared spectroscopic imaging is the imaging version of spectroscopy, meaning we have an instrument which gives us data of this sort, which is a data cube of this sort, where on the XY plane here, the image plane, you get an image just like a microscope image, but additionally you get the third dimension of data. So for every pixel here, you get a spectrum, and this is the same spectrum, the mid-infrared spectrum that I showed in the previous slide. So there is biochemical information encoded in the spectrum, and our goal is to look at every point, look at every pixel, look at the spectrum, and using the chemical information encoded in the spectrum, identify the disease state of a tissue. That's the essence of this technology. And this is the old technology, which is FTIR imaging, which FTIR spectroscopy or FTIR spectroscopic imaging, which many of you may be familiar with. And our goal is to use this approach and do diagnostics with it. And how do we do diagnostics? Here is the framework. This is a machine learning framework where we start with the data which we have collected, which is the data cube, and then we reduce it to a potential set of features or metrics. And so this incorporates prior information about the different molecular constituents that are expected in different tissue subtypes. Then we use machine learning algorithms. In this case, I'm giving you an example of prostate histology, where we are trying to divide tissue into different subtypes, and we can use different machine learning algorithms and do some optimization. And after a few iterations, we can come up with a potential set of metrics and statistics of how well the classification has worked. So we use a pathologist class annotation as the gold standard, and we have used a large number of algorithms, Bayesian algorithms, genetic algorithms, neural networks, random forests, CNNs, more recently deep learning, and all of them. It turns out that the combination of mid-infrared spectroscopic imaging and machine learning works very, very well. To give you some numbers, it gives you more than 0.99 or 99% accuracy as measured under the area under the curve of an area under the ROC curve for both prostate cancer and breast cancer. In breast cancer, I'm showing you an example where we have 7 different tissue subtypes over here, and we can get like about 94% cancer detection accuracy and about 99% cancer subtyping accuracy. And this has been validated extensively in my work and in others' work with over 1,200 prostate cancer patients and over 1,000 breast cancer patients. And this turns out to be remarkably robust. It is a very good way of doing analysis. So this was an FTIR imaging. However, there are some challenges with FTIR imaging in terms of imaging speed and resolution and other things. Those are the things that I'll talk about next. Okay, how do we address the limitations of this approach? Photothermal imaging is one of the more interesting and important infrared imaging techniques that has evolved in the past few years. And the important thing that it does is it addresses the problem of spatial resolution. For example, it gives us a 0.5 micron spatial resolution across the entire range of wave numbers. So in my lab, we have an instrument that works in the 800 to 1800 wave number range. And across the entire range, it gives us a half a micron resolution, which is much better than the 5 to 10 microns diffraction limited resolution of IR alone. And the way it works is that it uses a mid-infrared pump, which is the red beam shown here, which excites the sample, and there is a photothermal expansion, and there is a second laser beam, which is the green laser over here, which acts as a probe. And when the infrared is modulated and the visible light that comes back is demodulated, you get very high resolution. The resolution is determined with the green laser, and therefore it can give us much better resolution than the infrared, which is a long wavelength, much longer wavelength, something of the order of 5 to 10 microns, which gives us 5 to 10 micron of the order of 5 micron resolution. The usefulness of this spatial resolution in the context of cancer tissue analysis, and let me start with an example of ovarian cancer diagnosis. So traditionally ovarian cancer diagnosis is done with this process. So you take a biopsy out, then what's called a needle core biopsy, then it's embedded and sliced into multiple sections, and then you would remove the embedding medium and then apply a bunch of histological stains. For example, you do H&E, which is the most common, and then you do a panel of different stains to essentially identify different molecules, and then a pathologist visualizes looks at the tissue under a microscope and makes a determination. What we want to do essentially is replace this pipeline by something more quantitative and something which can be much more repeatable. So there is a significant variation from pathologist to pathologist when this kind of an analysis is done, because it is subjective. So if the same doctor or pathologist looks at the exact same tissue under a microscope, the diagnosis that come up with can be as much as 20 to 30% different. And so this variability is an issue, and all the different tissue processing steps, cutting, slicing, staining, and all that, we want to have something which is robust, which gives us a robust analysis of the tissue. And one thing we can do is, instead of using all this panel of stains, we can just use infrared imaging, which has which identifies all the molecular information in these panel of stains and gives us similar information without actually needing to stain this kind of tissue. Additionally, we use machine learning or AI methods to do the interpretation, which will be the same in any hospital. Wherever you go, it will essentially be we'll get identical results. And additionally, infrared is quantitative. That allows us to make the quantitative information from infrared plus the machine learning gives us reliable and robust and repeatable results. That's the basic idea here. To give you an example of what the data looks like, so this is the traditional H&E stain here, image of, these are 2 ovarian cancer patients. So this is FDR image, and you have photothermal imaging data from my lab over here, and you can see that there's much higher resolution compared to the FDIR over here. And we have done this kind of an analysis for about 100 ovarian cancer patients. It's a tissue microarray, and if you want to zoom in to illustrate what we can see, so here is an example of the raw photothermal imaging data we get over here, and this is the H&E, the stained images. So first of all, notice that there is a good correspondence between the yellow regions here and the purple regions here. It says that our infrared image gives us molecular contrast without actually adding any contrast agents or stains or any kind of chemical. The other reason why this is important is, if we use machine learning, then what essentially machine learning does is quantifies this qualitative insight that, oh, these colors look the same as these colors. It quantifies this idea. Like in machine learning, a big problem is that you have garbage in, garbage out. If you just read it a lot of data, you can get kind of spurious, like hallucinations, all kinds of results, which kind of may look reasonable but actually are false. And so ensuring that there is a correspondence between these kind of infrared images and the H&E, this is kind of ground truth, ensures that what we are doing is reliable and that we can trace it back to a molecular constituent or a bunch of molecular groups, which is a very important thing in medical diagnostics. Whether something is reliable, how did you arrive at the answer, what is the molecular basis for making a diagnosis, is a very, you can't just say this is the diagnosis. We should say, why is this, based on what is this diagnosis, what is the underlying kind of molecular change that we are identifying. And IR, infrared imaging, gives us the ability to do that, which is a big advantage when if you want to translate infrared to the translate machine learning to the clinic. So here is like ROC results for 78 cancer patients. You get about 98% cancer tissue subtapping accuracy for epithelium and stroma. So this is a very robust and reliable result. With other methods that we have tested, we got much lesser kind of not as good results. And one of the things to notice with this approach is that so the high spatial resolution has a big advantage. So with just 5 wave numbers, instead of collecting 300 wave numbers in FTIR or other methods, you can just collect 5 wave numbers in the spectral region. But unlike FTIR, we have much higher spatial resolution. So we are trading off spatial and spectral resolution. Higher spatial resolution, but fewer wave numbers. And so this kind of trade-off was previously not possible, but now it is. You can only collect a few wave numbers, which improves the data collection time quite dramatically. And additionally, you can get very good tissue subtapping accuracy. And we use this, and we also came up with a metric called stromal ratio, because we have quantification of the on every pixel. We can find we found that compared to normal, even grade 1A, 1B, 1C patients, there is a significant reduction in this particular metric called stromal ratio with p less than 0.01. So this is essentially like a biomarker for ovarian cancer. I wanted to give you another very closely related result, which is cervical cancer. So we have another data set of 100 cervical cancer patients. We did a similar analysis and got an accuracy of 0.94, and we have further extended this study to 290 cervical cancer patients with multiple samples from each patient, and we get very similar results. So this is ongoing work, and the results are extremely promising. I wanted to switch gears a little bit and talk about another technique, which is called dual comb spectroscopy, which addresses a slightly different problem that FTIR has. FTIR takes time to collect data. Is there a way to speed it up quite dramatically by orders of magnitude? And here is one approach which works quite well. This is something we have been working on for the past several years, and we recently have gotten very good results, which I'll show. The basic idea behind this dual comb spectroscopy itself is known, but applying it in the context of photothermal and in the clinical context is something which we have pioneered. So the way dual comb spectroscopy works is the comb. So there are these things called comb lasers. So you have a laser where at different wave numbers, at uniformly spaced wave numbers, have a bunch of laser lines. Basically, it's one laser which emits at a uniformly spaced wave numbers. You take one laser, take a second laser where you stretch the spec comb a little bit. So you take the blue one, take the red one, which is slightly stretched version of this blue laser, and interfere the two. So there's a beat frequency at each wave number, and this beat frequency, unlike in the optical frequencies, the beat frequencies in the RF, it's something of the order of kilohertz to megahertz range, and this can be measured with electronics. So basically, there's a one-to-one correspondence between this beat frequency and the wave number here. So in that way, basically, you may put a detector here if there's an absorbing medium, measure the spectrum before and after the absorbing medium, and you get the spectrum, absorption spectrum, in a single shot across all different wave numbers in a single shot. This is the basic idea. So you can do, instead of one wave number at a time and sweeping a quantum cascade laser or single wavelength laser, you can get like 100 to 1,000 fold improvement or even more in speed with this kind of an approach. So we did that. In fact, we built one of these systems and attached a microscope to it to do imaging. And first thing is, even with 500 average, it just takes something like 38 milliseconds. And to give you some context, it takes about 0.1 to 0.2 seconds, or up to even 1 second to sweep across the spectrum. You can do it in milliseconds and up to microseconds for the entire spectrum. So that's the improvement in the speed you can gain. we did imaging with this, and here is a USAF target where these bars are like a polymer on calcium fluoride, or I believe it's silicon slide, an SU8 pattern on a silicon slide, and you can see that you can get very good spatial information and very good spectral information over here, and very fast. We also extended this to ovarian cancer. This is ovarian cancer tissue, and we get spectra at different points, which looks reliable. And this is in the 3000 wave number, 2900 wave number region, 28 to 2900. But we can get this kind of information at different wave numbers. So this this imaging method gives us information, but it's still diffraction limited. The next thing we did was combine photothermal imaging with dual comb imaging. So what does that allow us to do? It gives us the high spatial resolution of photothermal, which is something like half micron, but the speed improvement of dual comb. So combining the two, we can get high speed as well as high resolution compared to all previous infrared imaging methods. And so this is essentially how we did it. So we have like a CW laser and a mid-infrared dual comb laser. One comes from the top, one comes from the bottom. Combine the two and have it incident on the sample, and basically you have each wave number is modulated at a different frequency, and you get a signal at the photodetector, and you essentially demodulate it. The way you do that is take a Fourier transform of the data, and each modulation frequency corresponds to a different wave number, and this way you can get information across multiple wave numbers. And this happens to work really well. And so here is an example of here are a few examples. We have a polystyrene bead, and we can see that the reference spectrum matches quite well with the photothermal infrared spectrum, and this is a spatial resolution is also quite good. And this is a 5 micron bead, and you get something like 5.6 to 5.9 microns, which is very good resolution as well. And we did it on an SU8 polymer, and in this wave number region, and this works very well as well. So this is one, and this is very current and ongoing work, and this allows us to do get advantages of both methods, and much faster data collection and much higher resolution than previous methods. I wanted to talk about some of the interesting applications, clinical applications. So this is myelofibrosis, which is a bone marrow disorder, where in the bone marrow you have these black what are called reticulin fibers, which are about half to 1 micron in resolution, which are infiltrate. And what we want to do is identify the direction and orientations of these fibers, and there is currently no good way to quantify this. And so here is the kind of image you get with conventional FTIR imaging and QCL based imaging, and this is the photothermal. You can see that it's much better resolution, and you can see much higher quality data. And so here is like an example of where this is a reticulin fiber. You can see it's half to 1 micron resolution, which you can see with just a visible in microscope, or with photothermal you can see the fiber, but you cannot see it with FTIR because the resolution is not enough. So first we can see something which you could not see before. Second is we can quantify the orientation. So this is useful. So another thing we did was, in my lab we introduced polarization contrast into photothermal imaging, and we can get important insights. So we have a paper on this which you can look up from a couple of years ago. And so if you introduce polarization contrast, you can essentially quantify horizontal fibers are right here, and in this case the direction is minus 45 degrees here, and direction is 45 and 0 here. So different orientation of fibers can be identified and quantified using some tensor calculus and with this polarization contrast. And what we do after this tensor analysis is get images of this sort, where you can look and observe the orientations of different fibers and the locations of these different fibers. And this is important because as the disease progresses, these fibers become crisscrossed, and when there is low-grade disease, they are parallel. So by quantifying this fiber orientation, we can track the progression of the disease and see if the therapy which is been given to a patient, is it working or is it not working? You can answer these kind of questions. That's the basic idea. The last clinical example I'll give you is lupus nephritis, which is a kidney disorder, and these are the filtration units of the kidney. And the problem with is that there are no good biomarkers for accurate diagnosis of lupus nephritis, and we are combining infrared imaging with traditional histology to come up with much better biomarkers, where infrared imaging can add molecular contrast, and we have come up with several biomarkers which we think will be exciting. To wrap up, I hope I have convinced you that mid-infrared spectroscopic imaging is a very powerful technique, especially for digital histopathology, and it has a great potential for clinical translation. It can be used in the clinic quite extensively, and we have new techniques for photothermal imaging, dual group spectroscopy, which are very promising, which solve a lot of the problems of previous infrared methods. And I've given you examples of bone fibrosis and lupus and even ovarian cancer, and I think this is a very promising field of research, and it will make a big impact in the clinic in the next few years. With this, I would like to thank some of my students and collaborators and my funding sources, and thank you for listening.
Thank you, Rohith, for that fascinating talk. I would now like to introduce our final speaker, Belinda Pastrana. Belinda Pastrana, PhD, has more than 30 years of experience in molecular biophysics and biochemistry with a comprehensive understanding of protein development and characterization. Currently, she is the Chief Scientific Officer, co-founder, and board chair of Protein Dynamic Solutions, Incorporated. She is an I-Corps graduate and has been awarded the highly competitive NSF SBIR grant. She has a postdoctorate in molecular biology and pharmacology from Mayo Clinic and holds PhD in biophysics and biophysical chemistry from Rutgers University. Dr. Pastrana is also a tenured faculty from the University of Puerto Rico campus with over 280 peer-reviewed publications, abstracts, granted patents, and scientific presentations. She is also an invited author of a book chapter in the Encyclopedia of Analytical Chemistry and has numerous awards, among them the Henry Dreyfuss Teacher Scholar and the Distinguished Woman in Chemistry and Chemical Engineering in Puerto Rico, as well as a track record of training the next generation of biopharma professionals. Belinda, thank you for joining us today. You have the floor.
I would like to thank the organizers for inviting me to give this presentation to you today, the title of which is The Potential for ICHQ5E End-to-End Comparability Assessment Using a Streamlined Screening Platform Method for Multiple CQAs. The topics that I will be presenting will include an overview of therapeutic proteins and the International Council for Harmonization, known as Q5E for biologics, an introduction of developability and end-to-end comparability, the quality by design of our protein mentor to be fit for purpose for the ICHQ5E comparability exercise, and through a case study be able to present our comprehensive rapid screening method for multiple attributes, the assessment of aggregation, and how the protein dynamics can confirm differences in stability. I'll also provide a conclusion and the acknowledgement. The inspiration for the design of therapeutic antibodies has been addressing the different mechanisms of action. And with that, the architecture has evolved dramatically. Yet the analytical tools to be able to characterize these therapeutic proteins has lagged behind. The reason why therapeutic proteins are complex is also due to the critical quality attributes that need to be assessed, and there are many. I'm highlighting in green those that I will be discussing today due to the impact of formulation optimization or formulation conditions. And this is observed in both developability as well as end-to-end comparability to ensure product quality, safety, and efficacy. And those are protein aggregation, the concern for self-association, especially under high concentration, high dosing conditions, and the stability of the therapeutic protein and its formulation condition. The ICH-Q5E is a global guideline that allows for the comparability exercise to be designed to evaluate the risk of an adverse impact on the regulated drug product. We need to ensure the product quality, safety, and efficacy. That strategy is implemented under the following areas: during extended characterization, any process control, during an update of a process control strategy, and during release testing. Now that we understand the mindset and the reason behind this regulatory global guideline, it would be to think about the strategy of implementation. When do these comparability exercises are performed? So one would be during a new manufacturing site evaluation. You would have data from batches from an existing site against the new site. Another would be new equipment or new instrumentation being implemented, so data from an old or existing equipment against that of the new equipment. A process change, so pre and post change samples or data is then evaluated as well within this comparability exercise. Another would be a supply chain, so material from the original supplier against that of a new supplier. Another situation would be during the investigation of a deviation in manufacturing. This comparability exercise would involve sampling at multiple points during the bioprocess and evaluating them. And finally would be a batch-to-batch comparability exercise, which would be a routine operation. We believe that our case study best matches this last exercise. But you can always think about developability, formulation optimization, and then having that same thought process and method be implemented at different points during the bioprocess, downstream bioprocessing. Or just prior to having the drug substance become the drug product. So in the next to last formulation condition to the final formulation stage. The drug life cycle can be separated into two categories. One being the developability phase. This is when the therapeutic protein is subject to physical chemical characterization. The IND and BLA filings are performed, and then you also have the end-to-end comparability assessment, which is basically once that therapeutic candidate has been approved for commercial use. That is the time when the drug-regulated product is subject to the global guidelines. And so you have the 21 CFR Part 11 compliance, you have the risk mitigation, and we also would need to consider those product quality attributes that have been defined. And in 30% of the cases today, this involves high concentration, high dosing, subcutaneous administration. And that also needs to be considered. So even though the drug life cycle is defined in phases of the exploratory research, preclinical research, pilot study, and manufacturing, in the case that I will be presenting today, would be ideally suited for when formulation optimization and then the release testing evaluation is done. But that does not preclude it from also being implemented during the pilot scale phase and in the multiple stages or phases of the bioprocessing. So this platform method has been fit for purpose to be able to take the journey with the therapeutic protein throughout its entire life cycle. So today's presentation is going to do a deep dive in terms of the evaluation of formulations and their role on the stability of a therapeutic protein. Thinking about developability, the evaluation involves the formulation optimization. You want to be able to evaluate the following CQAs: self-association, secondary structure, any aggregation events, and the stability of the therapeutic protein under the different formulation conditions. But then think about the end-to-end comparability. It would be ideal to be able to use a very similar method whereby one can ensure the product quality, safety, and efficacy. So those same CQAs that I mentioned above, and being able to ensure that the formulation conditions are the correct ones, especially for certain excipients, if they can be detected prior to the release testing. We wanted to explore what would an event of protein aggregation using this new 2T2D correlation algorithm would be represented as. And so with the case that we published this year in Applied Spectroscopy, using other two-dimensional correlation algorithms, we have been able to reproduce the unique feature of this positive peak on the top triangle of the symmetrical plot, which has an unusual feature of the side of the palm of your hand. This is representative of a deamidation event that led to aggregation of the protein. The next topic that I would like to expand on is that of the quality by design of our protein mentor platform and how it's ideal for the ICH Q5E comparability assessment exercise. The opportunity to de-risk preclinical candidate selection within the developability assessment, and then the end-to-end comparability assessment following the ICH Q5E to ensure the regulated drug product quality, safety, and efficacy is ideal, especially when one considers that since the 1980s, only the AMID1 region using IR was the focus of the study of evaluating proteins. The other aspect is that today, not only by the use of the QCL microspectroscopy, but also the ability of being able to do the data analysis using two-dimensional correlation analysis, and there are multiple algorithms that we're employing on this regard, then we can expand the evaluation to looking at those surface exposed glutamates and aspartates within the protein, as well as the AMID2 band, where we're focusing on those side chain interactions and how the loss of stability of those weak interactions can impact secondary structure and also the stability of the protein. The two-dimensional correlation algorithms allow us to monitor in real time 11 of the 20 amino acids, as well as the secondary structure. If I were to compare CD and fluorescence to our protein mentor platform, which is the green bar, is that it's capable of evaluating that side chain mode. or side chain in general. And red is that it does not. So our Protein Mentor platform allows us to be able to enhance the spectral and temporal resolution. And that is what is key to be able to perform the analysis that we do in such a comprehensive manner where the relationships between the CQAs is understood. And so the first 11 residues that I've listed here are polar residues as well as the aromatic residues that can easily be monitored using fluorescence and CD. But with that are the weak interactions that govern stability of the protein and interactions between the therapeutic antibody and its antigen. And indirectly, we can monitor the effects of disulfide scrambling because those weak interactions would be perturbed and the architecture of the therapeutic protein would collapse. But IR is extremely sensitive to secondary structure changes, and with that, the sensitivity to aggregation. Providing now all of the enhancements necessary to dive in and to understand at the molecular level those key critical quality attributes to ensure product quality, safety, and efficacy. What is unique to our Protein Mentor platform is that of the slide cell array, because it allows us to be able to do comparability assessment, even following the ICHQ5E, where, for example, 3 different batches from an originator site can be compared to 3 batches from a new site and have that be in triplicates. And so that's part of what we are providing, but the more important aspect is that that comparability assessment for multiple CQAs is done at 25 degrees and in just 10 minutes for either 6 samples in triplicate or, in the developability case, 18 individual samples. We can accelerate speed to market and also de-risk preclinical candidate selection during developability. And the reason being is because of our platform being able to take advantage of multiple 2-dimensional correlation spectroscopy analysis. That is the basis for the evaluation of both the preclinical candidates and the drug-regulated products. To date, we can evaluate multiple CQAs in a slide cell array for up to 21 candidates. That also includes the epitope map. The greater the number of CQAs that are being evaluated for an intact therapeutic protein in its formulation condition, the lower the risk in your selection. But it's also very important to understand that there is a comprehensive analysis that is being done, as well as a quantitative analysis that is being reported. Clearly, the value of this platform method is to be able to evaluate the intact therapeutic protein without any tags or labels under varying formulation conditions, and comparing that with the final formulation, which would be ideal, especially in the case of release testing, where it is very probable that the conditions are under high concentration, high dosing conditions. I would like to now bring to your attention the case study, which involves adalimumab. As a typical drug substance in a phosphate buffered saline solution at pH 7.4 versus what would be the drug product in the original formulation at pH 5.2. So this only involves two spectra that are evaluated at 25 degrees for the amide-1 and amide-2 regions. The complexity of the formulation conditions is very different. The phosphate buffered saline solution is at pH 7.4. There are no excipients. While for the original formulation, you actually have a combination of two different buffers, the phosphate and the citrate in saline at pH 5.2, with excipients being the polysorbate 80 and the mannitol. So the complexity of these two buffers is dramatically different. And we wanted to evaluate what did these two formulation conditions do to stabilize this therapeutic protein. For example, the solvent accessibility, surface area, the weak interactions, and how these weak interactions stabilize the structure. Is there a propensity for aggregation, and the general stability of the protein? Let's first evaluate solvent accessibility. And for these evaluations that I mentioned earlier, we're using one correlation algorithm that's called 2T, 2D, or two-trace, two-dimensional correlation spectroscopy, whereby the weighted difference of the spectra is subtracted, and we then, by the use of these two-dimensional plots, increase both the spectral resolution, and we can also separate the contributions of each protein sample regarding its spectra. And they are symmetrical in nature. So this first panel, panel A, shows what would be the results with the blue crosspeaks. This is an asynchronous plot. Those are the contributions of adalimumab under the pH conditions of the phosphate buffered saline solution at pH 7.4. And this yellow feature with a series of concentric contributions would be the adalimumab in the original formulation. And so I am summarizing those absorbance differences when we compare the two spectra. And the standard deviation provides the level of statistical robustness in this comparison for n equals 3 samples or evaluations. On panel B, I'm actually summarizing the results obtained for the asynchronous plot in panel A, along with the quantitative report in the table as a bar graph. And the error bars would provide the summary of that standard deviation. The assignment of these cross-peak coordinates is always going to be defined or described in a lower gray shaded area for everyone to evaluate and to see. But basically what we're doing is dissecting this plot. And we can go ahead and focus first on the solvent accessible surface area. So here we're looking at both glutamates and aspartates that are surface exposed, that are hydrogen bonding with the aqueous environment. And so the greater this solvent accessibility and exposure to the aqueous environment, then there is a higher risk for the self-association events. And also, there could be potentially a higher risk for degradation events such as deamidation. The relationship between the solvent accessibility surface area is due to the weak interactions with the aqueous environment. And those differences can be obtained by looking at the region within the 2T2D plot that I'm highlighting. So those coordinates are summarized in the table, and we obtain that, not surprisingly, in PBS at physiological pH, there's a greater number of weak interactions that are being observed. Regarding the secondary structure differences, then within the region that I'm highlighting now, there's greater secondary structure contribution at physiological pH as compared to the original formulation condition. We can now consider the 2T2D plot in terms of colloidal stability because we're looking at differences in formulation. But now taking it a step further. For example, within this spectral region, we can now be able to establish the side chain modes in their locations regarding the secondary structure. And that allows us to be able to establish how these formulations stabilize the secondary structure, as well as the weak interactions that are on the surface of the protein. You have to think of those two items. On the next slide, I will confirm those sites using the amino acid sequence and a structural model of the Fab regions of adalimumab. In the comparative analysis where we established the differences between adalimumab in the two formulation conditions with a simple spectral comparison, we can now be able to bring that information over to the structure of the protein within the Fab regions. And those amino acids located to specific secondary structure would apply to these select sites that we have identified. And they're all surface accessible, so they're all being potentially impacted by the formulation conditions and the colloidal stability as well. The interaction with the excipient and how it stabilizes the secondary structure or not. Adalimumab does not exhibit any aggregation under any of the two formulation conditions evaluated today. I'm also showing the small icon that summarizes what would be a typical aggregation plot using 2T2D. In the case of evaluating thermal stability for the entire thermal ramp, which would be 24 degrees to 56 degrees, we're using the co-distribution analysis that allows us to be able to establish which therapeutic protein under which formulation condition is more stable, relatively speaking. And so it's simply due to the number of cross peaks. And to be able to distinguish from the 2T2D, I'm using the lower plot for the co-distribution. And in this case, the positive cross peaks would be perturbed prior to the negative cross peaks, so red and yellow prior to the light blue and dark blue regions. There's yet another two-dimensional correlation algorithm called 2D costs. The plots are not being shown today, but the analysis of that plot allows us to determine the protein dynamics. In this case, what are the differences in the protein dynamics under the two formulation conditions? But I also have the opportunity at this time to show you the reproducibility of those dynamics for two samples on the same slide as they are being heated. In this case, in the presence of PBS on panel A. The close relationship between those two symbols is stating the reproducibility of the measurements. At the same time, for the original formulation, the closeness of the overlay is substantial, considering that these are the changes that are occurring due to the dynamics of the protein. Once again, two different samples on the same slide in the same formulation condition. So now when I show you the comparison of adalimumab in PBS at pH 7.4 with that of the therapeutic protein in the original formulation, we can safely establish that the weak interactions are being stabilized that are surface accessible by the formulation and the excipients in the original formulation. And precisely the blue circles within the region of 31 to 36, which is that sequential order, the longer it takes, then the more stable it is. On the other hand, we can also evaluate the beta sheets, the helical regions, and the loops of the protein in comparison to the two different formulation conditions. And once again, you see the later phasing towards a later moment in time under the original formulation condition, suggesting that they are also being stabilized. Not so for the beta turns. And that is fine because it is the beta turns that are mostly responsible within the Fabs, within the CDR regions, that would interact with the antigen. In this slide, I'm showing the full spectrum of 1800 to 1000 wave numbers that allows us to be able to then focus on evaluating the glycan region of the protein, as well as any excipients, and confirming their presence in the formulation. What are the effects of colloidal stability in terms of pH differences, and also the contribution of citrate on the AMA2 region. And the same for the polysorbate. So this relationship can be established using the entire spectrum. In this next slide, I'm now presenting the 2T2D for the full spectral region. Remember, on the lower left-hand side was the main evaluation of SASA, secondary structure, amino acid composition, the weak interactions, and how they were being stabilized or destabilized by the formulation conditions. In the full spectrum, I can now confirm and validate peak assignments, but also establish those cross peaks that are associated with a specific excipient. And one example is the mannitol, highlighted in panel A with the arrow. There are 3 cross peaks, all that are positive, so they're associated to the original formulation. On the table, I have a bracket highlighting at 1550, 1082, 1034, and 1424 are all cross peaks associated with the mannitol, which is also highlighted in the bar graph in panel B. This is just an example of the comprehensive nature of the evaluation. The results presented provide the basis for having proven the usefulness of this platform method in a scenario of developability and end-to-end comparability assessment. The system allows for a comprehensive understanding of the physical-chemical properties and the relationship between CQAs. The potential for its implementation following the ICHQ-5E guidelines is evident to ensure product quality, safety, and efficacy. I would like to thank spectroscopy, as well as the Koblenz Society and the Society of Applied Spectroscopy, the National Science Foundation for providing the funding to develop this platform, Dr. Saunoda, who has described for the first time in these two-dimensional correlation algorithms, and our sponsors. Thank you.
Time to present during today's morning session. As a reminder to everyone in the audience, you can submit a question by typing it in the Q&A box, which you can find at the bottom of the screen. Once again, please type in the name of the speaker your question is for. It is now time to begin the question and answer session with today's speakers. So let's start with the questions. Well, thank you, Belinda, for that excellent talk. And thank you to all of our speakers who have taken the time to present during today's morning session. As a reminder to everyone in the audience, you can submit a question by typing it in the Q&A box, which you can find at the bottom of the screen. Once again, please type in the name of the speaker your question is for. It's now time to begin the question and answer session with today's speakers. So let's start with the questions. Let's start with Rohith. How does photothermal imaging compare to Raman imaging?
Thank you for the question. Photothermal imaging is, so infrared and Raman are two complementary ways of getting vibrational spectroscopy information. One of the big advantages of photothermal is that the efficiency of photothermal imaging is much higher in the sense that Raman, as many of you may know, is an inefficient process, relatively speaking. If your million photons are incident, like only a handful will come back as Raman photons. But IR is much more efficient, like it's several orders of magnitude more efficient in terms of the process itself. And therefore, what it means in practice is that photothermal imaging can be much faster than Raman imaging.
Another question for Rohith. How fast can dual comb imaging collect a spectrum?
Dual comb can collect a spectrum, one spectrum in something of the order of 76 microseconds or so. So it's very fast compared to give you a sense, to compare this, to get a full spectrum from a standard quantum cascade laser, you need about 0.1 to 0.5 seconds, so half a second. And this is like 76 microseconds. So it's several orders of more than 10,000 times faster in terms of one sweep of a laser.
Another one for Rohith is, can polarization photothermal be used in pharmaceutical analysis?
Absolutely, yes. So there are big advantages to photothermal. being used for pharmaceutical analysis. For example, one of the things that I mentioned in terms of polarization contrast that you can achieve with photothermal, it allows you to see materials which are chiral, for example, where in pharmaceutical analysis, when there's chirality involved, polarization photothermal is a very good way of dealing with those kind of samples. Additionally, you can go to the sub-micron length scales, and you can see the microscopic structures in pharmaceutical analysis, and that could be useful in a lot of applications as well.
Well, let's switch over to some questions for Belinda. Will your protein mentor system provide the advanced testing necessary to manage the complexity of the new therapeutic proteins in the pipeline?
Sorry, I can't hear.
Belinda, you must be on mute.
The answer is, can you hear me now? Sorry.
Yes.
The answer is yes, because we are looking at the amino acid level resolution, especially being able to probe at the surface of the protein and looking at those hydrogen bonding interactions within glutamates and aspartates provides that empirical information that is lacking using other techniques. So if you think of NMR having a high resolution atom level understanding of protein dynamics of the full intact protein, in this case, we have the same, but looking at the functional groups. So that allows us to be able to, in real time, understand protein dynamics and the interaction with excipients as well. So it's not only just looking at secondary structure, we're actually understanding the mechanism of aggregation, if there's any self-association at high concentration, high dosing conditions, is an area of need currently in the biopharma industry, and we can address it.
Well, thank you for that. Another for Belinda, is your platform technology readily transferable to manufacturing? And that's a yes or no, and why?
The answer is yes. And the reason being is because we're not using any tags or labels, no kits. So that allows us to have a platform method that is transferable from developability or R&D directly to bioprocessing. And that's the short answer.
All right, another for Belinda is, what does your system provide that other techniques cannot?
Being able to understand the relationship between different CQAs directly, because we're monitoring these multiple CQAs in real time for the intact therapeutic protein, where we've streamlined many of the standard operating protocols for sample preparation. The other aspect, let's say, for example, you're interested in evaluating whether how stable is your protein, is this aggregated between two different formulation conditions, between 2 or 3 different chromatographic steps, then our technology would be ideal to be able to evaluate such event.
Another for Belinda is, have you received any feedback from the regulatory agencies?
The answer is yes. They very much agree that the system is potentially valid for the application of the ICHQ-5E global guidelines of comparability assessment. They are very keen in being able to see how the comprehensive information can be tied to the statistically relevant and robust results that we can provide.
And another for Belinda, why do you define your system as a multi-attribute platform method?
We can in one set of spectra be able to evaluate, for example, is there any deamidation in the protein? Does that deamidation event lead to aggregation or self-association in protein? And be able to relate those results to the stability of the protein and the protein dynamics, like I showed earlier today in the presentation regarding the protein dynamics.
This one I can throw out to either of our speakers here is, how do spectroscopic techniques contribute to efficiency of biopharmaceuticals in terms of cell viability or drug efficacy or drug release?
If you want, Rohit, I can start.
Yes, please go ahead.
In terms of cell viability, for example, I would say the extent of glycosylation, because we know that the cell culture conditions can drive to the assurance of having the proper glycosylation of the protein. And any variations there can lead to variations in the glycan profile of that therapeutic protein, putting it at risk to being stable and having some sort of immunogenic event, which is of concern. The other aspect is always the aggregation throughout the bioprocessing event is something that is always a matter of concern. So any test that can be performed that has a fast turnaround time without having to heat the sample is always desirable.
Another question. Oh, go ahead, Rohit.
Rohit might want to answer as well.
I just want to answer it quickly, very quickly. So traditional infrared imaging for doing it in live cells was actually quite difficult. But one thing we have done recently with the photothermal version of infrared imaging is that we can do live cell imaging and tracking of drugs as a function of time. So that's something which is just in the context of photothermal and IR imaging has become possible just recently. I just wanted to point, yeah.
And here's another question that's just addressed to either speaker. Is there perfect strategy and methods for therapeutic proteins? And because some of the activity of proteins, such as enzymatic activity, proteins can be denatured by heat, how do you resolve such problems in your therapeutic process? And are there ways of measuring this?
Infrared is highly sensitive to secondary structure changes. We're using on cascade laser microscope, so it's extremely fast, and we can actually monitor these changes in real time as they are occurring. We don't need to do a thermal heating event, but we can use it as a risk mitigation step in developability to make sure that that therapeutic protein is manufacturable, and you won't have issues of aggregation or self-association due to the formulation conditions or temperature conditions of the protein. So our system is ideal to be able to have that predictive nature of understanding the stability of the protein overall, evaluating multiple critical quality attributes for an array of samples. So lead candidates can be evaluated, and that has the effect of de-risking preclinical candidate selection.
Here's another question that's not addressed to any particular speaker. It says, how do you measure weak interactions of proteins? Do you think zeta potential and free energy measurements have an impact on protein dynamics?
Zeta potentials are, as I understand, model dependent. And in our case, we do not fit our data to any specific model, none whatsoever. And it's a priori no knowledge is needed. So we're actually just monitoring those weak interactions in real time. Disruptions of salt bridge interactions, the extent of hydrogen bonding, like I showed in this presentation today. That allows us to really understand those colloidal stability issues that might arise, and be able to correct with using the optimization of the formulation condition via adding certain excipients like the mannitol or an arginine or histidine as an excipient in the formulation conditions. They all add to the stability of the protein.
Here's a general comment. Great talks, with many exclamation marks. And here's another question that could be for either speaker. As I was curious to know if your techniques can be used to assess the manufacturing quality of mRNA vaccines.
The answer is yes. We have done several feasibility studies. I am not at liberty to discuss them, but not only looking at messenger RNA and the double-stranded content, but also the lipid nanoparticle and the messenger RNA encapsulated lipid nanoparticle form. So all of those on the same slide cell array, and also do the thermal perturbation to evaluate stability, for example. So everything that I've discussed regarding proteins can be applied to messenger RNA, DNA, and lipid nanoparticles because we have the spectral region of 1800 to 1000 wave numbers, which allows us to be able to really understand these different components within such a complex mixture.
Well, with that, let's wrap up this session. We want to thank the excellent speakers that were invited to present today. And we want to thank the audience for attending and for participating in the last morning session of this virtual symposium, Emerging Trends in Pharmaceutical and Biopharmaceutical Analysis, presented by Spectroscopy, the Society for Applied Spectroscopy, and the Covalent Society. Thank you again to our sponsors, CEM, PerkinElmer, Thermo Fisher, Metro, and Anderson Hauser. We also want to remind you to take advantage of the resources from our sponsors, which you can find in the bottom of your screen. You can still sign up for the afternoon session in this virtual symposium using the link in that resources section. If you would like a certificate of attendance for this event, please send an email within a week of the event to training@s-a-s.org. That's training@s-a-s.org. You will receive an email alerting you when this virtual symposium is available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. We'll now break for lunch ahead of our afternoon session. We hope you'll join us as we reconvene at 2 p.m. Eastern Time, where we'll hear leading instrument suppliers give educational 20-minute talks addressing applications, tips, and best practices in the use of vibrational spectroscopy. We also hope to see you at other sessions where we have more great lineups. Well, goodbye for now.
Thank you.
Thank you.
Thanks. Those were excellent. Thank you very much.